Here we go. Well, welcome everybody back to the award ceremony. I am Nicole Theodosiou as chair of the Professional Development and Education Committee. It is my biggest honor to introduce the winner of the Viktor Hamburger Prize at the annual meeting. The Viktor Hamburger Prize for Education recognizes individuals who have made outstanding contributions to education in developmental biology or related fields. I am thrilled to announce and introduce this year's winner, Dr. Joe Handelsman. Dr. Handelsman is the Willis Research Professor and Director of the Wisconsin Institute for Discovery at the University of Wisconsin-Madison. She was previously on the faculty at Yale and is also a Howard Hughes investigator. It would be easy to recite a list of her accomplishments at the bench. However, today I will highlight the equally important scholarship contributions she has made to undergraduate education in biology. Over the past 10 years, Dr. Handelsman has written countless instructional materials, including Scientific Teaching, a quintessential resource for all educators, and Entering Mentoring, a guide for being a thoughtful mentor to students. In addition, she has served for many years as the editor of the journal CBE Life Sciences Education. Dr. Handelsman has been a major influence in the teaching style of many, using teaching as a way not only to hone the students' scientific insights, but to make them educator consumers of information. Through her position as director of the Scientific Center for Scientific Teaching at Yale and the National Academy Summer Institutes on Undergraduate Education, numerous instructors have been trained and mentored by her. Dr. Handelsman has also been an advocate of women's issues in science. Her 2012 study, published in PNAS, showed that faculty, when given applications of students with randomly assigned male or female names for lab manager positions, the faculty rated male applicants as significantly more competent and hireable, gave them higher starting salaries and more career mentoring than the identical female applicants. The analyses also showed that female students were less likely to be hired because they were viewed as less competent. This work redirected efforts to advance the participation of women in science. Her contributions to science education have been previously recognized by the American Society of Microbiology, Nature Magazine, and the American Institute of Biological Sciences. In 2011, she was awarded the Presidential Award for Excellence in Science, Mathematics, and Engineering Mentoring by President Barack Obama. President Obama later called on her to serve as the Associate Director for Science at the White House Office of Science and Technology Policy, advising the President on ways to change and improve aspects of science, in particular STEM education. Dr. Handelsman has truly made exceptional contributions to the field of biology education and all STEM education nationwide. It is my honor to award Dr. Handelsman the Viktor Hamburger Award in Education. And thank you for joining us today.
Well, thank you, Nicole. I appreciate your wonderful words, and this award is an amazing honor. I hope that something that comes along with it is the longevity of Dr. Hamburger, because he lived to 101, and that sounds pretty good to me. These are difficult times to talk about anything but racism and COVID-19. But I'm going to talk today about some of my recent efforts in science education. And I can tell you, as I'm sure many of you have experienced, that recent events have changed the way we teach and what we teach in this venue. Both racism, which has now become part of the curriculum, and ways to avoid racism and how to be an anti-racist have become topics that are standard in this curriculum now, as well as ways to teach when you can't go into the classroom, which for a research-based course is kind of challenging. So this last semester has had some pretty dramatic effects on the topic I'm going to talk about today. So I'm going to tell you the story of Tiny Earth, which is a method of crowdsourcing science with students as the scientists. And I'd like to start with the reasons that we developed this curriculum in the first place. So the first is a report that based on a report that I helped write, actually chaired a committee that wrote for President Obama in 2012. And one of the things that we found in the course of exploring that report was that the workforce is wholly inadequate for STEM. We have a severe lack of STEM literate students graduating from college, as well as STEM majors. And this has been kind of lost on the scientific community because of all the debates about whether we have too many PhDs or not enough PhDs. Nobody's really been focusing publicly on the bachelor's level. And it turns out that quite soon we'll probably reach the 1 million mark in the size of the workforce deficit for STEM fields. So this is a serious concern to all, should be for all STEM educators. And one of the concerns that we had in writing this report was that some of our best students leave STEM during college. And so we need ways to keep the good students in STEM. Once they've declared an interest, those are the easiest ones to recruit to STEM because they've already expressed an interest in STEM. We just scare them away with our teaching methods. Another challenge that we all face is that there's just so much information to teach. Anyone who's taught for more than a few years knows that we're constantly in the mode of deciding what not to talk about, because as we add new information to our courses, we need to delete older information. And so our education system has to be efficient. And most of us are looking for more efficient ways to teach. We also know from surveys of employers of our students that they need to be able to deal with the intellectual challenges of employment that they're likely to have after college. And that includes the ability to think analytically and creatively. And of course, if they go on to graduate school, these are the same skills that they'll need under those conditions as well. So we propose that we need a global shift in teaching methods, and that these teaching methods should be evidence-based. And Nicole mentioned the book, Scientific Teaching. That's the topic of the book, is using science in our teaching to make our teaching more effective, and using the evidence base, which now is quite powerful and robust, to choose our teaching methods and design our courses. So what are evidence-based teaching methods? Where do they lead us? These are methods that will hopefully improve learning and student retention, improve grades and performance, and also improve the inclusiveness and diversity of science. And these methods have been shown through copious experiments and studies about how people learn and why they leave science, to include active learning. And one of the points that we mentioned in the report that we wrote for President Obama was the incredible power that research courses have. These are courses that you can offer to freshmen and sophomores before they might get into a faculty member's lab to do their own independent research. And the reason they're so powerful is that when students are taking their introductory courses, which are often the very courses that drive them out of science, if they have a research experience, they become inspired and excited about science, and it gives more meaning to the book learning that they're doing. So those are the educational reasons that I started Tiny Earth in 2012. But it also dovetails with another interest of mine, and that's the antibiotic crisis. And this is one of my research interests, and it's also a very personal interest because my mother died of antibiotic-resistant bacteria that could no longer be treated with any known antibiotics. So I have several drivers driving my interest in antibiotic resistance, and that became the focus of my educational interests as well. So what is the antibiotic resistance crisis, and why are we concerned about it? So probably most people have heard that bacterial pathogens are increasing their resistance levels to many of the antibiotics that we have at our disposal. And some of the worst pathogens now have multi-drug resistance and simply cannot be treated. But at the same time, and this is the disturbing part, we have very few new antibiotics coming down the pipeline. And this is because the large pharmaceutical industries pretty much abandoned antibiotic discovery in the 1980s. And here you can see, on the one hand, the sharp increase in resistant bacteria. These are antibiotic-resistant infections in people. But the blue bars show you the release of new antibiotics into the marketplace. And since the 80s, which was probably one of the peak times for discovery and registration of new antibiotics, we've seen a steady decline, and it's even lower at the present time. So at the very time that we most need antibiotics to treat bacteria, we are not generating them from the drug industry. So why is that? We now have only 3 companies of the major pharmaceutical companies, Merck, Roche, and GlaxoSmithKline, continuing to do the discovery part of antibiotic study and marketing. And so the question is, why is that? The pharmaceutical industries found that antibiotics are essentially too effective to be profitable. We take antibiotics for a few days to maybe at most a month for a very severe infection, and then we stop. Whereas chronic diseases are much more profitable because the drugs are taken usually for the rest of a person's life, or at least long term. The discovery process is hard and it's expensive, and without the payoff of long-term use drugs, that balance just doesn't pay for the companies. But one of the things that the companies said in the 1980s when they were leaving this field is that the soil is tapped out. There's nothing left to discover. They said that 99% of the antibiotics you find in the soil will be old antibiotics. And I'll come back to that point. So other companies besides these big 3 do sell antibiotics because it turns out it's very profitable. But they basically reformulate and rename, sometimes chemically modify, but usually just reformulate versions of old drugs. And this industry is currently $45 billion. It's projected to go up to $60 billion by 2025. And the CDC predicts that antibiotic resistance will be the leading cause of death by 2020. So this is not an unimportant source of income for pharmaceutical companies, but they've just found they can make more money by investing in other kinds of drugs. One thing I think it's worth pointing out in the current environment is that in the 1918 flu epidemic, most people who died actually suffered from bacterial infections that followed the flu. Their systems were weakened, their lungs were already infected, and it was easy for bacteria to get established. And this is a common effect with flu and many other respiratory infections. And so the bacteria were actually the cause of death. And it's interesting that we had 2 subsequent pandemics of flu in 1957 and 1968. And I bet nobody remembers those. I certainly didn't know about them the way I've always heard of the 1918 flu epidemic. And the reason is that many fewer people died because antibiotics were abundant and available during these later epidemics. We don't yet know what the role of bacterial infections are in the COVID-19 pandemic. It's known that about 70% of patients who are hospitalized end up on antibiotics. So we may not be hearing about bacterial infections because they may be treated before they arise. But I think we'll learn more in the coming years about whether antibiotics played a role in this epidemic as well. So Tiny Earth is an undergraduate introductory level course in which students are tasked with discovering antibiotic-producing bacteria. Most of the antibiotics on the market today come from the soil, from soil bacteria, and so that's the source that the students use. I started this when I was at Yale in 2012. I had 6 students sign up, so I thought, well, this isn't going to be very successful or popular. But today it turns out to be quite popular. And at the end of the course, and I'll come back to this, the students can send their bacteria that they've isolated to our chemistry hub at the University of Wisconsin, and we will do further characterization of their isolates. So the goals of this are to inspire students about the power of science. We teach them about this global epidemic of antibiotic resistance and how the companies are not filling the need, and how the students themselves can play a role in solving this problem. And that's very exciting to them. They also find it very exciting that they're part of a worldwide network. And I didn't know that that was going to be true. That was something I only discovered after talking to my students at Yale. They may have been few, but they were passionate about the course. And I asked them, so if students all over the world were taking this course, would it increase or decrease your excitement? And I was very surprised to find out that that was a major part of their excitement, and they loved the idea of being collaborative or just side by side with students all over the world. It teaches them an enormous amount of biology because antibiotics inhibit necessary cell functions. It gives you the opportunity to teach just about every function of a bacterial cell, and then to explain the difference between the toxicity to bacteria and eukaryotes or people. It requires going into eukaryotic biology as well. So we have actually used this as an introductory first semester biology course, because you can work all of the topics of a general biology course at the cell and molecular level into the curriculum. And the ultimate goal on the industry side is to make antibiotic discovery less expensive and a lot faster for the pharmaceutical companies. And that takes proving to them that it can be done, because many of them are still living with the paradigms of the 1980s. And not only has education changed since then, but the science has changed enormously. So this is an indication of how fast things happen, whether it's epidemics or educational change in this day and age. We started with this tiny group of students in 2012. And now last year's count was, we believe we had about 700 instructors trained. We have a training course that trains instructors. 300 of them we've trained in the United States, and then our collaborator in Spain has developed Micromundo, our sister initiative, and he has trained 344 instructors in Spain and Portugal. So we now have probably about 10,000 students taking the course every year. We know the instructors because we've trained them all. There are about 700 of them, but it's harder to get numbers for the students. And you can see this is now a worldwide network. A few points are missing in here that we haven't quite filled in yet, but we're moving to more and more international sites across the world. So what do the students actually do? So the lab techniques are, first of all, they collect a soil sample, and they don't really put it in their hands like that. We actually do teach them good sterile technique. They then take that soil and they dilute it in a buffer or water, and they spread a drop of that on solid media and see what grows up. And that in itself is an education, because they're shocked that all of these gorgeous and varied colonies come out of just a simple old soil sample, usually taken from outside the building where we teach, or outside their dorm, or in their backyard. They then isolate the bacteria from this kind of zoo of organisms and get them into pure culture. And then they test the individual isolates for the ability to inhibit one of 6 what we call escape strains. These are the so-called escape, because those are the acronym that the Infectious Disease Society of America has designated as 6 of the top antibiotic-resistant threats in the world. We also use a couple of eukaryotes. Sometimes some instructors use yeast, some use a protist called Pythium. And others use other targets, but these are the ones we generally provide for our instructors and that they use to test for antibiotics. And you can see this is a plate that's been spread with one bacterial strain. This was a staph strain. And then these are the individual isolates, and the ones that produce a zone of inhibition around them must be producing something that inhibits bacterial growth, and that would be an antibiotic. So of course, the big challenge is how many of these antibiotics are actually new, or are we just rediscovering old compounds? And that's where the Tiny Earth Chemistry Hub comes in. The students usually end just after this step. They usually do some taxonomic analysis on the producing bacteria to find out what their 16S ribosomal RNA gene sequence says about what they're related to. Some of them do chemical extractions of the cultures to see what the solubility of the compound responsible for inhibition is. But about that time, usually the semester ends. And so the instructors are invited to send any isolates they'd like to our chemistry hub, where we are developing and using new methods for discovery of antibiotics, and particularly to separate known antibiotics from new ones. And so why are we arrogant enough to think that we're going to discover new antibiotics in soil that the big pharmaceutical companies missed? And I've been asked that very question by big pharmaceutical companies. And the answer is manifold. First of all, we use new screening methods. And so I won't go into them, but we know today a lot about why bacteria express or don't express antibiotic biosynthetic pathways. And so we have all sorts of tricks of culturing them with other organisms or stressing them that induce them to produce antibiotics. We didn't have those tricks in the 1980s. So the industry was not looking at the same set of pathways as we are. Also, we're looking at new targets. Acinetobacter is also known affectionately as Iraqibacter because it was discovered during the Iraq War. It was first discovered in the deserts of Iraq and infected soldiers. Now it's pretty much ubiquitous. We find it in hospitals and all over the United States. It causes very serious infections, but it wasn't available, it wasn't around. We didn't know it even existed as a pathogen in the 1980s. So we found, for example, some antibiotics that are specific for Acinetobacter. They will only inhibit Acinetobacter and no other bacteria that we've tested. Well, clearly the antibiotic industry would not have found those antibiotics because they weren't using Acinetobacter in their screens. We also find that the students innovate enormously. Every time we teach this course, we find something new about screening that the students have discovered, and we have many, many different methods that we now use based on student discoveries. And the most important probably tool that we have is the prioritization process in the chemistry hub. So the pharmaceutical industry has said that 99% of the compounds we find will be old compounds. And with our volume, that's actually okay if only 1 in 100 is new, and we have 10,000 isolates coming through every semester. That's a reasonable number of new compounds. And we actually don't think there's great evidence for this 99% number. But the question is, how could you get rid of the 99 and only focus on the one new one? And that's the challenge that we can only address based on the last few years. If we could spend the time, which is the really time-consuming part of doing the purification and then determining the structure of the antibiotic only on new compounds, we could make our discovery process incredibly efficient. And anybody who's done this kind of work knows the frustration of doing months of purification and then structural analysis and finding out, as we did a few years ago, that you have citric acid and not a new compound. So what's our process for finding out which organisms are producing new compounds and which are producing old ones? So this was the first round of isolates that we analyzed from students. We got 3,000 isolates from the students. We found 1,500 had the particular activities that we were most interested in, so we pursued those. And we're collaborating with the Joint Genome Institute at the Department of Energy's Lawrence Berkeley lab that does massive genome sequencing for us. Out of the first 46 genomes, we found 394 pathways, and I'll tell you a little bit more about their newness in a second. We then also look at the metabolome, and just like in genomics, where you can look at genes and determine based on known genes whether the genes you're looking at are related to antibiotic biosynthetic genes, you can do the same now with metabolomic profiles. So we obtain a metabolomic profile and use the software that enables us to compare our compounds in crude mixtures with the databases of known compounds. And then we can focus on purification and structural determination only on ones that look new. And there were 3 underway structurally when COVID-19 hit, but we don't have structures on those. But we hope in a few months we will. So this is an analysis, an example analysis of what we use BIG-SCAPE, which is a network for analysis of all the nodes of biosynthetic clusters. The biosynthetic clusters that are similar will be connected, and then the ones that are different, or like the ones you see here, seem to be unique because they have no similarity to known clusters. Those will end up not being in clusters. And you can look, you can see here, this is from the first 46 genomes. The red ones are only found in the Tiny Earth database, and the black ones are found in known organisms and known clusters. So there's a lot of new genetic material here. There are a lot of biosynthetic clusters. BGC, by the way, stands for biosynthetic gene clusters, which are recognizable because bacteria do this nice thing of clustering all the genes needed for synthesis of molecules in a single operon. So we can look for a long string of biosynthetic genes. These can sometimes be as much as 20 or 30 genes, as much as a couple of hundred kb. They're enormous, but fortunately they are recognizable. So in these first 46 that we analyzed, we found 178 new or total gene cluster families, so these kinds of clusters. We found 22 of those had known relatives, but 156 of the families were previously undescribed. And so this is a remarkable amount of newness and novelty that we can find in this database. So this is an example. This is a strain that we got from Wingate University students. And we got the full genome sequence and found that, in fact, there's a 100% match to a siderophore biosynthetic pathway. That's an iron binding compound, and a terpene that looks like a carotenoid kind of compound. So those are ones we would ignore. Obviously, that's exactly what we want to weed out before we do the purification. But if you look at these two, there are no known relatives. We can recognize some of the individual genes and assign function, but there are no known relatives of the pathway. This is one, we have a few high schools in the network, and this is one that was isolated at Glenbard East High School. And it's a strain of Pseudomonas that has 9 biosynthetic gene clusters. And one of them is particularly interesting. This is the layout of the cluster. You can see that we can identify many of the open reading frames and their likely functions. Some are resistance genes, making the cell, the bacterial cell, able to produce the antibiotic without killing itself. Some are core biosynthetic genes, some are regulatory, so we can lay out that cluster. But what's really exciting about this particular region, 3.8, you see here, is that this looks like an NRPS terpene hybrid. So antibiotic biosynthesis has often taken 2 different kinds of pathways and jammed them together. This is a novel kind of motif, and we're really excited about having discovered what may be a new motif in antibiotic synthesis as well as a new antibiotic. We then couple that with the metabolomic analysis, and this is the same strain from Glen Bardis High School. And when we look at the families of metabolites by LCMS, MS, that's MS2, and then the GNPS, this is the San Diego's metabolomic software, we can begin to group our isolates and their metabolites in families again. And some of these are known, some of them it's very hard to find relatives. You can see at the bottom, they have no known relatives. Some, as you can see, fall into known antibiotic families, but their actual structures differ from the known members of those families. And so this gives us a clue about what kinds of compounds might be in the strain. And in this case, we interpolate the data between the biosynthetic gene cluster that we find by genomics and then the chemical families. And in this case, we have a candidate that looks like it might be the NRPS terpene hybrid compound, and that will be one of our very first targets for purification. So that gives you a sense of where we're going. I apologize that we don't yet have structures, but COVID-19 surprised us all. And soon we're hoping to have a demonstration of the power of this, this pipeline for discovery, be able to obtain a frequency of new compounds versus old ones to bring to the pharmaceutical industry and hopefully rejuvenate that industry by helping them make discovery more efficient and less expensive. These are the members of the lead team in the Tiny Earth Network. Nicole Broderick is the head now, actually at just moved to Johns Hopkins University, is the leader of the training and has done all of the training since the very early days. Sarah Miller is the executive director of Tiny Earth, and all of the rest of these great undergraduates, graduate students, and postdocs have contributed to mostly the chemistry, the genomics, and also training students and instructors in the technology and techniques of Tiny Earth. And of course, the Joint Genome Institute is responsible for the genome sequencing. They've been incredibly generous to us and enabled us to develop this pipeline the way it looks today. So I'd like to thank not only my collaborators here that you see their names, but all of the Tiny Earth instructors. These are the ones that came to our last 2019 symposium, the last one that was in person, as well as the many, many thousands of students across the world that are contributing to the data in Tiny Earth. And with that, I thank you for listening to a bacteriologist who's not exactly a developmental biologist and allowing me to join your meeting for the day and awarding me this very humbling award. With that, I'll take questions if there are any.